We may not have a football team, but, that was kind of weak. Come on now. All right. Oh, yes. Well, I see this is going to be a lively crowd today. Thank you. All right. So they tell me that there's a rumor going around. They tell me that there is some spectacular research going on at UCSF. See, I like her. Look at that. She must have been a GEPA student. Okay. So today, we are going to continue the theme that we have in the morning session, and we're about to hear some spectacular research that's going on at UCSF. So thank you for being here today, and of course I'm excited to hear from this year's incredible lineup of speakers, and therefore, even though I have already added to my introductory remarks, they're going to be short. The Discovery Talk, as many of you know, because many of you have come up to me and said how much you enjoy this session, is the Graduate Education and Postdoctoral Affairs signature event at Alumni Weekend. It gives us the opportunity to highlight some of the exciting science, research, and discoveries that are coming out of UCSF. Now, I wouldn't be a graduate dean if I didn't talk to you a little bit about our programs, so I'm going to talk to you a little bit about our programs, if that's okay. So, at the moment, we offer 20 PhD programs in basics, translational, and social population sciences. We always forget our soc-pop folks, but they are here too. 13 master's degree programs and 2 professional doctorates, one in physical therapy and one in nursing practice. And might I just ad-lib again? Our DNP was recently recognized as #1. All right. Today, we will hear from UCSF faculty with their lightning-fast discovery talks about breakthrough science and innovations that are changing the world, from advancements in neuroscience, AI and drug discovery, global precision medicine, the microbiome, DNA, protein behavior, and cellular regeneration. Like last year, we also have 2 trainee presentations. You're gonna love these. So each year, students at UCSF vie for a competition that we call Grad Slam. Basically, they have to, in 3 minutes, talk about their research in layman's terms. Try that at home. But this year, you are in for a treat. And let me tell you why. Because last year's winner, there's a UC-wide competition. So all the campus winners go up to Sacramento, and there's a UC-wide competition. And wouldn't you know, for the first time in our history, UCSF won. And today, you will get to hear her talk. Folks, hang on to your seats. You're about to go for a dive into discovery. Ladies and gentlemen, let me present to you all the speakers today.
Reza Abbasi, AI and the Brain, How AI is Driving Our Knowledge of the Vast Universe Inside Our Minds.
Hello, everyone. So what I want to talk about today is the intersection of these 2 perhaps exciting fields, neuroscience and AI. And many of you may be using AI when you use your smartphone or smartwatch, or when you interact with a chatbot. But the type of AI that I'm referring to here is what I call science-friendly AI, a tool that we can use to understand super complex systems in nature. And my research team here at UCSF really sits at the intersection of these two subfields under neuroscience. Basic neuroscience, where we try to understand the foundations of how brain works, and clinical neuroscience, where we try to understand neurological and psychiatric disorders. And here's the challenge. This system is a super complex system, our brain. It contains roughly 86 billion neurons, which makes it very, very hard to study, and also very expensive given the cost of current experimental tools out there. But we have to start somewhere. So we start here, mouse brain. 70 million neurons is still super complex, but within reach. And many of the principles that we learn from mouse brain, we can scale them up to human brain. So here is a fundamental question in neuroscience. What are the regions in our brain? What's the map of the brain? What are the functions of these regions? And this is the kind of question that I'm very interested to use AI to answer in my research team. So what we did, we took large-scale whole brain genomic data sets from mouse brain, and then we built this AI tool, what we call Cell Transformer, and then we fed this entire data into the AI system to make sense of it all. What came out is one of the most detailed maps of the brain ever created. So I'm super excited about this, not only because of the resolution, but also because of what we found when we explored these regions. So we are able to redefine some of the known brain regions with higher precision, but more importantly, we are able also to discover some new regions and subregions in the brain that do not exist in current maps of the brain. So for example, that dark purple area that you see up there, it's a brain region in this particular slice of the mouse brain. It's a brain region called midbrain reticular nucleus. It's known to be involved in controlling arousal and integrating sensory and motory signals, but the organization of this region is mostly unknown. So we looked up our map, and then we found that we are able to identify these 4 subregions here, colored in different colors, with unique genetic profiles. We are now working closely with biologists to validate them, actually name them, and understand their functions, further characterize their genetic profiles. So that's nice, but that's just part of the story, right? What perhaps is a deeper question is how these neurons interact with each other, how they drive each other, and how they influence each other. And that's a key question in neuroscience because many of our functions, such as cognition or decision-making or memory, is really emerge from these chain of reactions between neurons. So to answer that question, we built another AI tool. What you're about to see is 1 millimeter cube in mouse brain. It's just size of a grain of sand, but containing hundreds of thousands of neurons. So using our AI system, we're able to identify more than 160,000 connections in this particular region. And what made us excited about this is that many of these sort of trends of connections actually follow and is consistent with real wiring diagram of the brain, how neurons are actually connected anatomically together. We are now working on understanding how these connections are disrupted in some of our neurological diseases, such as Alzheimer's. So what I've shown you so far is how we use AI to understand these questions on the basic neuroscience side. But similar AI tools could be used for us to explore patient data and to understand neurological and psychiatric disorders in that level. So just some examples. We can use AI to now direct and guide deep brain stimulation therapies in disorders such as chronic pain and Parkinson's. We have now AI integrated in a smartphone and smartwatch to help us track and diagnose movement disorders such as Parkinson's, just using, in this case, 3 to 5 seconds of video from cameras. We are also able to integrate AI with biosensors These are sensors that measure the stream of physiological activity from brain and body to better understand emotion, mood, and predict how these emotion and mood are disrupted, perhaps in psychiatric disorders such as depression, and also perhaps a science-friendly version of large language model that we can use in clinic to guide clinical decision-making. So across all of these projects, we have actually open sourced all our AI models, so anyone, anywhere on the planet can use them to look into their own data from brain or body. And all of these are available on our website. Just to wrap, I do believe that brain is one of those very complex systems, maybe the most complex system in nature. And perhaps, maybe for the first time in the history, we just might have the right tools to start to understand this very complex system. Thank you.
Please welcome to the stage Joanne Chun with her presentation, How Fast Are Your Drugs? How Computational Discovery is Bringing Drugs to Market Faster and Why It Matters.
Hi, everyone. I'm super excited to be here today. My work here at UCSF focuses on educational innovation. So thinking about how we train our scientists for fields that are rapidly changing. And this is especially true in drug discovery. So today I'd like to share how AI is transforming the way we discover and develop drugs and what that means for the future of medicine. So developing a drug today takes more than 10 years and can cost over $2 billion. And even after all this time, around 90% of drugs fail during development. So despite all the time and effort, yes, we are still needing to do something. So here, we're going to talk about how AI can help transform each one of these steps in the process of drug development. So we're going to start with finding a target. So before we find what to target, we have to understand disease. And for conditions like Alzheimer's, cancer, diabetes, there are such complex biological systems at play. With thousands of genes, proteins, and pathways interacting with one another. And traditionally, scientists would study these systems piece by piece. So one gene, one protein, one pathway at a time. But today, we're working with massive data sets that capture all of these interactions at once. Far more data than any one person can analyze. And this is where AI becomes very helpful. AI can scan through these large data sets and find patterns that can allow us to pinpoint which genes or proteins might be driving a disease. And once we know what to target, the next step is designing a drug. And traditionally, this involved testing thousands and even millions of compounds in the lab, hoping to find something that works. Today, a lot of that work can be done computationally. We can leverage AI models to generate and evaluate new molecules and help predict the ones that are most likely to work. So instead of testing everything in the lab, we can use AI to help us prioritize the compounds that are most promising. Next comes safety, which is one of the biggest reasons a lot of drugs fail. A lot of drugs look promising early on, but prove to be toxic later in development. AI can help us predict this much earlier. By learning from previous data, AI models can predict or identify patterns that are associated with toxicity. This allows us to filter out drugs that can be risky before they ever reach patients. Next, we go into clinical trials. This is where drugs are tested in humans. And this is also the most complex and expensive part of the process. Here, we can leverage AI models to help us identify the right patients for the trials, help us optimize drug dosing regimens. AI models can even help us design better trials. And even after the drugs are approved and marketed, the work doesn't end. We continue to monitor how these drugs perform in real-world settings. Here, AI can analyze patient data and help detect rare side effects and improve the way we use drugs over time. So this ongoing learning helps make treatments more effective and safer. So as we can see, AI has the potential to transform every step of this drug discovery process. But that also creates a gap because the science is advancing faster than we can train people to use these tools. So we created a new program, a new master's in artificial intelligence and computational drug discovery and development. This program trains students at the intersection of computation and biology. Our students learn how to code, build models, but most importantly, they learn how to apply these tools meaningfully to real-world problems in drug discovery. And what's really exciting is seeing what they're already accomplishing, how they are approaching problems in entirely new ways, and asking, how can we do something different with AI tools? And this is just the beginning of what's possible. We are at a moment where science and medicine are changing so quickly. But one thing that is certain is that AI will not replace scientists, but scientists who can leverage AI tools will transform the way we treat disease. And the people who will be leading this transformation are being trained right here at UCSF. Thank you.
Teresa Kortz with Global Precision Medicine, leveraging science to save children's lives.
Good afternoon. So imagine this. A young girl comes into the hospital. She's febrile, but her hands and feet are cold. She's breathing quickly, and she's abnormally tired. For the clinicians in the room, you might recognize these as ominous signs of a severe infection. If this child comes into Benioff Children's Hospital across the street, we would perform a risk assessment, initiate a rapid diagnostic workup, and we would start infection-targeted treatment. But many children in the world actually present to hospitals that look more like this. And while the child who presents to Benioff Children's is likely to survive, the outcomes here are actually very different. So I'm a pediatric critical care physician, which means a major part of my job is actually dedicated to saving children's lives. And while I work clinically at UCSF, my research focuses on global child health because unfortunately, geography is one of the biggest risk factors for mortality. Let me show you. So this is a world map. It shows child mortality. In the dark blue countries, mortality is 1 per 1,000 children will not live to their 5th birthday. But in red countries, it's 30 times higher. We wanted to better understand life-threatening or critical illness in children in these countries. So we did a large global study that included 19 countries and more than 7,500 children. And what we found was an association between country resource level and mortality. So the lower the country resources, the higher the mortality. We also found that 1 in 8 children presenting to the hospital already had acute life-threatening illness at the time of presentation. And in the most resource-constrained settings, that was as high as 1 in 3. It can be very challenging to do a risk assessment in these children based on clinical signs alone to identify the child that's at highest risk of death. We also found that 7 out of the top 10 causes of critical illness in children were due to infections. But unfortunately, only 1 in 10 of those cases ever has a definitive pathogen identified because of diagnostic capacity limitations. So as a clinician, this leaves you guessing as to what the patient has and what the best treatment is. So to that, we asked ourselves, how can we use the cutting-edge science that we have available to us at UCSF in order to address the challenges that clinicians face in resource-constrained settings? To that end, we did a cohort study in children with severe infection in Tanzania. It's a resource-constrained country in sub-Saharan Africa. And we measured markers of inflammation in the blood using a point-of-care device that could return results in 30 minutes with only a couple drops of blood. This is not Theranos. This device is already commercially available and approved for clinical use, I promise. When we took our best performing marker of inflammation, procalcitonin, and combined it with basic clinical signs, 86% of the time, we were able to assign a higher risk to the patient that died. Now, diagnosing infections is a little bit trickier because it tends to require tools. Microbial culture, PCR, antigen testing, which are not universally available, and in the case of culture, can be falsely negative if the patient receives antibiotics beforehand. So in this case, we applied a novel technology called metagenomic next-generation sequencing. And this is a very sensitive, unbiased pathogen detection method that works like this. So you take your patient who has signs of severe infection. We take a sample from that patient, and that sample contains both human cells as well as any pathogen that's present, whether it's bacteria, malaria, viruses, it doesn't matter, it's unbiased. In a lab, we then extract all of the genetic material, DNA, RNA, human or pathogen. That genetic material then undergoes processing and labeling so that it can be read by a sequencer. The sequencer reads millions of tiny fragments of RNA and DNA simultaneously. The raw data is outputted and uploaded into an open source, powerful bioinformatics pipeline called CZID. This then removes the human sequences, goes through multiple rounds of quality control, and then compares the remaining sequences to reference databases to identify all of the organisms within that sample. Now, every organism is not a pathogen, so we then pass it through a rules-based model to be able to identify the likely pathogen in this sample for this patient. Now, we applied metagenomic sequencing to our cohort of children with severe infection in Tanzania, and we were able to increase pathogen detection from 1 in 10 cases to 9 in 10 cases. And this is incredible, right? The one downside, well, there are many downsides, but big one is that it's currently not available in real time. However, the cost of sequencing continually is dropping, and there are initiatives that are specifically targeted at getting sequencers in low and middle income countries, so this may be a reality in the relatively near future. Well then, how do we translate these findings to help the patient that's in front of us? Based on the findings from this study, the National Referral Hospital in Tanzania has already updated their clinical protocol for severe infection in children. As a next step, we're testing whether patient gene expression signatures can actually predict type of infection so that we can translate that into a low-cost diagnostic tool. And that's how we want to take guessing to precision medicine, even in resource-constrained settings, because then we'll be able to do risk assessment that's based on the patient's own inflammatory response, combined with a tool that's designed for the pathogens in that location. And with that, we can deliver targeted therapy, and ultimately, this is what saves children's lives. So while geography often determines destiny, if we leverage the state-of-the-art technology that we have available at UCSF, where a child is born does not have to determine if they live or not. Thank you.
Please welcome Peter Turnbaugh with One Person's Trash, Why Poop Could Revolutionize Medicine.
Do you really want to spend your life studying poop? That's what people kept asking me when I decided to devote my life to studying the trillions of microbes that live within the human gut, called the human gut microbiome. When I joined this field, I realized that while we were learning a lot about how the microbiome affects risk of disease, we knew very little about what happened once the patient was diagnosed. And so I came up with the provocative idea that maybe the microbiome is actually the missing piece of precision medicine. Maybe differences in our microbiome could determine why one patient responds well to a drug and the other does not. We started our research in this area studying an old medication called digoxin. It's a natural product that comes from foxglove plants, and it's been used for decades to treat heart failure and cardiac arrhythmia. And while these flowers are beautiful, I would recommend if you see one in Golden Gate Park not to eat it because it's incredibly toxic. Digoxin has many side effects, and it's been known for years that microbes within our gut help to clear toxic compound from our body, forming a metabolite called dihydrodigoxin. But we didn't know how the microbiome does this. And this is not only important for medicine, it turns out to be really essential for culture. And so Van Gogh famously experienced a side effect of digoxin poisoning that makes lights look blurry. And that is thought to have inspired his painting of Starry Night. And so perhaps if Van Gogh had different microbes in his gut, he wouldn't have painted this. And so we sought out to find the gene responsible for digoxin reduction. We used RNA sequencing to scan the genome of the microbe that we know metabolizes this drug, looking through thousands of genes for genes that turn on when the drug is added. And we found these two genes next to each other that light up. We went on to use genetics and biochemistry to confirm that these are the genes responsible for digoxin reduction. And excitingly, we found that these genes not only predict whether or not single microbes in culture metabolize the drug, but they can also explain why which microbial communities from humans metabolize the drug. We also identified a way to interfere with this process by providing a dietary supplement, arginine, that inhibits the ability of the bacterium to metabolize the drug. But perhaps most excitingly, this provided a proof of concept for thinking about how the microbiome might affect other drugs and diseases. And so I was particularly motivated to think about cancer, given the famous and devastating side effects that occur in patients taking cancer chemotherapy. But the problem I kept running into is that I'm not an oncologist, and people don't want me to work on cancer. And so I did what many of you might think to do, and nowadays you might use ChatGPT. I used Google at the time and searched for researchers at UCSF that study cancer treatment. This led me to Alan Vinook, a member of the Helen Diller Family Comprehensive Cancer Center and a renowned researcher in the field of colorectal cancer treatment. His work throughout his career has actually changed how patients are managed today. Alan, for some reason, accepted my phone call, and that started a series of collaborations where we have studied many different ways in which the microbiome affects cancer treatment. One of the things we discovered is that just like digoxin, there are bacteria in our gut that contain an enzyme called pre-TA that inactivate the highly toxic anti-cancer drug 5-fluorouracil, reducing it to dihydrofluorouracil. Recently, we've shown in mouse models that if we elevate the expression of this gene, we can rescue mice from the side effects of cancer chemotherapy. And even more excitingly, we found that when we look at patients treated at UCSF that experience side effects of 5-FU treatment, we see that they have lower levels of the gene, indicating that this might actually be relevant to patient care. But we don't want to just understand why the microbiome affects treatment. We want to do something to intervene. And so we've been really motivated to develop new and more precise ways to change the genes and microbes that are in our gut. We use bacteriophage to target cells. These are viruses that infect bacteria. And then we deliver genome editing tools like CRISPR-Cas9 to target specific genes within these bacterial genomes. And this is part of the broader field of microbiome editing that's recently become emerging. We showed a proof of concept for this in mice, where we colonized mice with a strain of E. coli that contains 2 fluorescent genes, and showed that we could use a virus that infects that microbe to remove one of those fluorescent genes, changing yellow bacteria to red. And we're working very hard to extend these tools to look at other gut bacterial species, to improve the delivery methods, and to hopefully someday deliver pre-TA and other genes that we think might be beneficial for rescuing drug toxicity. And I'm very happy to be in the Bay Area, where we not only have excellent work being done at UCSF, but also across the street at Gladstone Institutes and across the Bay at Innovative Genomics Institute, and they're joining us in these broader efforts to target the microbiome. And so overall, our lab is very fascinated by the many ways in which the microbiome can affect drug response. And we hope to not just describe these associations between microbes and drugs, but to understand them and their cellular and molecular mechanisms. And while I have to admit the last couple of years have been really challenging to be a basic scientist and to try to maintain funding for our lab and all of these exciting research projects, but I do feel very grateful to be at a place like UCSF, where no one has ever questioned my commitment to studying poop.
Please welcome to the stage Postdoc Slam winner Atreya Day with his presentation, The Theory of 1,000 Grandmas. 100,000 grandmas.
When I was a child, I remember my grandmother used to love knitting, and something about the pretty colors and the soft texture of the yarn was so appealing that whenever I was nervous, I used to take out a ball of the yarn and played with it. Inevitably, the yarn would get all tangled up, and my poor grandmother had to spend hours untangling the mess I had made. Tangled string can be a nuisance for knitting, but did you know that tangled strings can also give you cancer? Okay, so the human DNA is a lot like a string. In fact, the DNA in the human genome is a 6 feet long string, and all that is tightly packed inside our tiny cells. And every time a cell divides, there's someone like my grandma sitting in the cells untangling the DNA. In the very rare case that a knot stays tangled, it can trigger conditions like Down syndrome or cancer. This tiny grandma untangling the DNA is a protein called condensin, and understanding how condensin does this is crucial for understanding these conditions like Down syndrome or cancer. So for a few years, experimentalists came up with a few clues to understand how condensin works. First, they found that condensin looks like a ring, and it can pass DNA through that ring like a loop by burning energy. The second clue they found was there are 100,000 condensins in a single cell. The third thing they discovered is what the whole 6 feet long genome looks like after a condensin has neatly packaged it right before cell division. So I am a theoretician, I'm a computational biologist. So using all these clues from experiments, I made this theory of 100,000 grandmas. Using my own grandmother as a reference, I assume each condensin molecule works like a grandma knitting loops out of DNA. When they get tired, they simply stop working and leave. When they come back, now they're grandmas. They don't remember where they were working. So they just start knitting at a random spot on the DNA. When two grandmas try to knit in the same spot on the DNA, they get into an argument. They both stop working for a while, but then they continue working as is. Using this theory of 100,000 grandmas, not only was I able to recapture the behavior of single condensin molecules, I was also able to get that bird's eye view structure of what the whole genome looks like. We believe this is the first step towards understanding condensin-related diseases. Now, it's been a while since my grandmother has passed. But even then, when I feel nervous, like when I'm up on a podium talking to an audience, it feels nice to know there are 100,000 grandmas in my cells protecting me from cancer. Thank you.
Please welcome to the stage Sophia Miliotis, Grad Slam winner, with her presentation, Finding HIV, a Swipe in the Right Direction.
5 years ago, after the worst breakup of my life, I did what any single millennial does. I got on Tinder. But if there's anything worse than heartbreak, it's getting trapped in an infinite scroll of tech bros, living in the cloud, but still down to earth. No matter how long I swiped, that feed never seemed to end. Funny enough, your immune system runs its own sort of dating algorithm, but instead of looking for love, it looks for a virus. Inside infected cells, major histocompatibility complex molecules, or MHCs, are like singles on a dating app. They're constantly swiping through tiny pieces of the virus, called peptides. If there's a match, MHC takes that peptide on a date to the surface of the cell. And there, the immune system checks in, like a skeptical best friend looking for red flags. And viral peptides are full of red flags, so once spotted, the whole cell gets destroyed. At least, that's how it's supposed to work. But some viruses don't play fair, like HIV, the virus that causes AIDS. It evades immune detection by rapidly mutating, generating millions of unique peptides the immune system has never seen before. And as a result, some infected cells escape and linger in the body for decades. But despite being incredibly diverse, HIV peptides are still on Tinder. They're still matching with MHC. And if we could pinpoint the exact HIV peptides that make it to the infected cell surface, we'd have a way to track down those lingering cells and destroy them for good. But here's the problem. Right now, we're simply guessing at which peptides might match with MHC and testing them one by one, and with millions of possibilities, we're stuck in that infinite scroll. My research changes this. I'm using a tool called EpiScan, which screens hundreds of thousands of HIV peptides at once. Here's how it works. I start with millions of engineered cells. Each one carries a single HIV peptide. Inside the cells, if there's a match, MHC heads to the cell surface. And the stronger that match, the more MHC shows up. So I can isolate the cells with the most MHC and check who MHC matched with. And that peptide may be the key to the next HIV therapy. Because finding love is a mess. But finding HIV doesn't have to be. Thank you.
All right, I need you to do your best football cheer. There is no doubt, thank you all, there is no doubt that UCSF is a very special place. Where else can you go on a single day to hear about AI in the brain, AI in education, global health and saving children, poop, and then talk about dating and grandmas? Come on. And all of this is based in fundamental science. A round of applause again for all of these participants. So if you hear the rumor that, not a lot of good science going on at UCSF, you know what to tell them. You know the stories now. Please share that story. Thank you so much for showing up today. Looks like we had a full house. We appreciate you. Now, all the speakers are going to be down here. I need you to come talk to them because they are doing some amazing work, and I'm sure you had questions. So come talk to them, ask your questions. Immediately following, I think it's at 3 o'clock, in this room, there's going to be the Global Health in Action and how we can advocate for science later on. I hope you enjoy the rest of your time here. I'm sure I'll see you around. Thank you again for supporting us. We appreciate you.
We do have some time, so if you would like to question the scientists, you may do so. If we want to do it in this format, I'm happy to pass the microphone to anyone who might have a question for any of our faculty members and current learners. If you guys want to take the stage, please. Thank you. Does anybody have any questions? Shy. Did you forget what you learned already? Okay, we've got a question back here. Thank you for being bold and brave.
Well, it's a silly question. I really appreciate all your presentations and what you're doing. Did you find a match?
I didn't find a match through Tinder, but I am happily partnered up right now. So. We met organically, if that helps.
So I have a question about grandmothers. This is important to me. What do you think the grandmother looks like in the cell? What exactly is it? A protein? Is it a...
Oh, it's a protein. It's like a 50 nanometer long protein. It's like kind of looks like a ring, and like the DNA can pass through that ring.
Any other questions? Okay, I've got one over here.
Yes, for our intensive care pediatrician, I was wondering, what is the cost of that procalcitonin test?
We can do a test for $8 per cartridge, and one cartridge does one patient.
Wonderful. And the other screening test, I know that's not quite available in real time yet. What's the projected cost for actually trying to identify the pathogen?
Yeah, there's the cost that we have, and then there's the cost that we hope to have. Right? So it still costs tens of thousands of dollars to be able to do that on this level. But we're actively working. I have a partner in Malawi who's actually developing the first sequencing lab in the country. And so again, we're like trying to get the technology closer to the source of the problem.
We've got another question back here.
There's so much that has been discussed kind of publicly about microbiome discovery, and that has not been part of many of our educational backgrounds that we've been exposed to. So could you just, I know this is a specific area that you're focusing on, but are there 2 or 3 themes that have emerged about microbiome and our well-being in the past few years that you might highlight?
Wow, yeah, that's a big question. So the microbiome does a lot, and I like to, compare it to the liver. If I were to remove your liver, probably lots of different things would change at once. And so the microbiome has its hands in many different aspects of the body. I think one kind of general lesson we've learned is that the role of the gut microbiome is not restricted to the gut. Traditionally we thought it was involved in infection in the gut and inflammatory bowel disease. But instead, we find that the gut microbiome can actually affect many different organs, including the brain, the lung, and others. So that's a very active and exciting area of study. I think that, it's hard to summarize a huge field. You gave me 3. I guess I'll use our own area. I think we have learned a lot that the microbiome does play a key role in the treatment of disease. And then finally, I think there's been a lot of progress in identifying kind of specific microbes and genes that drive risk of disease. And so that's provided new targets for intervention and is a really active area of drug development.
Okay, I've got a question here.
I had a question for the computational biologist. How did you, what is your background and how did you decide that this was the field that you wanted to go into? Because it sounds so interesting. I just read about like being mathematics and science together.
My PhD is in chemistry. But honestly, I have a PhD in chemistry because the professor I was working with was in the chemistry department. I mainly studied biophysics. Like I have like a physics minor, and like I studied biophysics. My background is like theoretical biophysics mainly, and I like working, like trying to apply like principle like Newtonian mechanics on biological problems. I think there's a lot of applications for that, especially with AI, because like we can make simplified models and then like simulate them with traditional mechanics and see how things happen, and they're really good predictions. The results that I showed today was based on that, but I didn't get into the details of that, obviously, of this talk.
We've got a question here.
This question is for the School of Pharmacy program. Is it interdisciplinary? Is it taught by people from the School of Medicine also? And in your program, do you take not even students, I mean professionals from medicine as well as pharmacy?
Yes, so we do have faculty from different programs in the school, School of Medicine, School of Pharmacy, graduate division, and we do take interdisciplinary students. We have some students who have a PhD, master's, bachelor's degrees as well.
I saw a hand up here, and don't worry, you in the front row, I'll come get you next.
This is for the poop presentation. So back in the early days of my nursing career, there was a physician that would order 72-hour stool collection. This wasn't a very fun job for the nurses, and probably less so for the lab that had to take it apart and identify it. Do you still do that? And if so, if not, what were the benefits of examining that much poop?
Oh, wow, yeah, we're really getting into it. What, are you guys eating? Yeah, that sounds wonderful to me, but I'm sure the patients wouldn't like it. There's been a lot of, it's kind of like inside baseball, but we have done a lot of work to like map the kind of biogeography of poop. And so, I think one of the reasons why your doctor was asking to do that is that, every stool is not the same as the next. And so, it's very useful to have kind of many samples from the same person. We typically don't do that on the same kind of 3-day window. We like to space them out a little bit more. And you get kind of the same ability to kind of average out differences within a person over time. But I have been involved in a couple studies where we were very interested in kind of the amount of energy from our diet that remained in the stool. And so there our collaborators actually did exactly what you said, and were able to not only measure the microbes in those kind of continuous collections, but the exact number of calories. And that provided one way in which we showed that manipulating the microbiome can actually change how well we digest our food.
Dr. Bennett.
Joanne, can you give an example of one of the projects that the master's students did with industry of what they discovered?
Yes, I think there are several. So we have projects that span the entire drug discovery process. So we have students who are actually using generative models to come up with de novo proteins, so new proteins that bind, right? So, and then we can also talk about projects that are at the other end of the pipeline, like using real patient data and analyzing patient data to see if we can gather any insights from that, like if patients are taking different medications and what are the interactions there, or different effects that can be picked up by just analyzing large data.
We have a question here.
You said that, again, for the development of drugs, you said that it costs roughly $2 billion from beginning to end to market a new drug and 90% fail. Your technology, can you have any projections on how much this will reduce the cost of developing drugs? And a follow-up question is, so will this actually result in decreased costs of new drugs to consumers?
We certainly hope that we can see a decrease in cost, but we've not seen those returns yet. And I think a big reason is because, again, we have these amazing, powerful tools, but not enough people who really know how to use the tools. So hopefully we'll start seeing those returns on investment in the next couple of years. So we're really hopeful that that will also help decrease the cost of bringing a drug to market.
Questioner.
First of all, your presentations were amazing and mind-boggling. So thank you for what you're doing. It's all, every one of you did great explanations of your work. Very inspiring. My question is a little more practical for our fecologist. What's your take on dietary fiber supplements?
This is not being recorded. Yeah, I would say I'm pro-fiber. I mean, I think it's hard not to be pro-fiber when you're in the microbiome field. It's probably, in my mind, the most well-studied aspect of diet that's been looked at. We know that many fibers are not digested by our human body, but are digested by microbes. And that plays a key role in both the growth of microbes in our gut, but also their production of metabolites that are beneficial. So, but I've worked a lot in the field of nutrition, so I also appreciate that many people experience severe side effects from fiber supplementation. And so, you do have to, kind of live your own journey, I guess.
And our final question right here for the day.
Well, I think Dr. Turnbau's work is obviously very popular, and I hope no one asked this question already. But perhaps relevant for many people in the audience, I don't know. There's a lot of interest in the GLP-1 inhibitors. Now, they have effects on the gut, obviously. And if you Google the question, are GLP-1 inhibitors good for the microbiome, it's all, positivity that you find on Google. But what are your thoughts about that?
Oh man, yeah. I think you're a plant, actually. I had that in a former version of the talk, but yeah, we have unpublished data that altering the microbiome or manipulating the microbiome can actually increase the ability to lose weight on GLPs in mouse models. And so we are very actively working on trying to understand the mechanism responsible and whether or not that is related to the primary mechanism of action of those drugs and changing feeding behavior, or whether or not it reflects other ways in which the microbiome affects energy balance. And so, yeah, I'd happily talk more about this after the session.
And with that, please give us a round of applause for our fabulous faculty and speakers. Thank you so much.